Welcome to the Cancer and Environment Forum 2022. What does new science say about chemicals and cancer? Lessons from public health and environmental medicine. My name is Tim Rebbeck, and I'm going to host the first session in this symposium. This workshop is sponsored by the Dana-Farber Cancer Institute, the Silent Spring Institute, the Lowell Center for Sustainable Production, the Zoo Family Center for Global Cancer Prevention, the Cancer-Free Economy Network, Mass General Brigham, and the Center for Cancer Equity and Engagement of the Dana-Farber Harvard Cancer Center. Welcome to all the participants and in this session. Many healthcare professionals have a difficult time keeping up with the role of environmental agents on cancer beyond the most well-established factors such as tobacco smoke or asbestos. Yet patients have a high interest in this topic because of fears of cancer-causing exposures and have opportunities to reduce their cancer risk by avoiding these exposures. Patients may experience healthcare providers' responses to their inquiries as dismissive sometimes. Healthcare providers may feel that they lack the knowledge to appropriately answer these kinds of questions. So we're presenting this forum series to better prepare healthcare providers to talk with their patients about cancer causes and to empower patients to minimize harmful exposures. To set the stage for this meeting, we're going to show you a brief trailer from a documentary about the experience of Dr. Margaret Kripke, the founding chair of the Department of Cancer Immunology at the MD Anderson Cancer Center and a former member of the President's Cancer Panel. During her tenure on the panel, Dr. Kripke and her co-panelists, Dr. LaSalle Lafall, dedicated their activities to examine the impact of environmental factors on cancer risk. Dr. Kripke will be speaking more about her experiences in session 3.
I went into this with a number of preconceived notions. I learned an enormous amount. I thought that all chemicals were tested before they were put on the market. Turns out not to be true. I thought that anything that's known to be a carcinogen would be regulated, also not true. And I thought that if there were regulations about something, that the regulations would be enforced, which is also not necessarily true. So that was a huge eye-opening experience for me.
So as Dr. Kripke said in the video, there's still a great deal we don't know about cancer and the environment. And even when we have data about exposures that cause cancer, we don't always act on this knowledge in a way that protects the public from potential harm. In this forum, we'll hear about foundational information about agents that cause cancer and some new science on cancer-causing agents. And we will hear a case study about childhood cancer cluster in Wilmington, Massachusetts. And this will illustrate how cancer clusters are identified and studied. In the first part of today's session, we'll have a presentation by 2 speakers. Following each presentation, we'll have a short question and answer. If you'd like to ask a question of the speakers, please type it into the Q&A area found at the bottom of your Zoom screen, and the speakers will answer as many of your questions as time allows. So to get started, I'd like to introduce our first speaker, Dr. Kate Guyton. Dr. Guyton is a toxicologist with expertise in carcinogen mechanisms, risk assessment, and cancer prevention. She's contributed to national and international evaluations of prevalent carcinogens such as trichloroethylene, pesticides, and tobacco smoke components. She's currently a senior program officer at the National Academies of Science, and prior to that was in leadership roles at the International Agency for Research on Cancer, the US EPA, and CCS Associates. Where she supported NCI cancer prevention projects. Her recent work has involved advancing systematic approaches for considering carcinogen mechanisms in order to better predict cancer hazards. So Dr. Guyton will speak on key characteristics of carcinogens. What are the tools we have now to identify carcinogens, including chemicals not considered to be complete carcinogens that may contribute to cancer? So Dr. Guyton.
Well, hello, everyone. I'm Kate Guyton. I'm very, very pleased and honored to be here today. I want to thank the organizers for this opportunity. I'm a senior program officer within the Board on Environmental Studies and Toxicology at the U.S. in Academies of Sciences, Engineering, and Medicine. And I'm going to speak to you today about some recent advances in identifying the causes of cancer. I'm going to reflect a lot about the work I did during my time at the International Agency for Research on Cancer, or IARC, which is part of the World Health Organization. So I just want to begin by declaring I don't have any financial interests related to the subject matter of my presentation. And what I'd like to do is really set the stage about cancer. What are the concerns of today and tomorrow? Talk about the progress that we've made in identifying cancer causes and also the promise for the future. And I'll spend some time talking about the key characteristics of carcinogens and what are some opportunities for providing new insights into the causes of cancer. So I'd like to just start with some data from the IARC, and you can certainly go to their website to learn more. Here are some data from 2020 and projecting over to 2040. We see a rising burden as well as changing the demographics. And prevention remains the single most effective response to these challenges because cancer carries an enormous burden for patients, for families, as well as financially and otherwise. And the first step in cancer prevention is to identify the causes of cancer. And this is done through authoritative review processes. I'm going to talk about the process that's used at IARC. It's not so dissimilar from processes used around the world. The IARC Monographs program is celebrating its 50th birthday this year, so it's been around longer than the US EPA has been around in this business of generating authoritative reviews. But you can also think to the US FDA and similar agencies around the world that do these authoritative reviews, and they provide the scientific basis for action. What are these actions? Well, there can be regulation to reduce exposures. These are taken by governments and address exposures in the workplace, in the home, in the environment, but there can also be actions by individuals. And I just want to emphasize that the path from evidence to action, it can be long, it can be steep, and there are many steps in the process. And I'm really going to be down in one of these very first steps, which is the hazard identification. Once this is done, there can be dose response analyses, and there can be other considerations before action is taken. So I just want to emphasize to you, this is just one step in the process. So how are carcinogens identified? As I mentioned, the Monographs has been doing this for a long time and periodically convenes advisory groups. This was done in 2018 most recently to update their method, which is called the preamble. And at the time that this was published, I made this word cloud, and I thought this would help to really emphasize that the basis for these evaluations is really about evidence that an agent causes cancer. And more specifically, it really comes down to three lines of evidence. And the first is cancer in humans, and these are epidemiology studies. I'm a toxicologist, so it's always slightly dangerous when I talk about epidemiology, and I will defer to the other esteemed epidemiologists you'll hear later. heal from later. But there's also data on cancer in animals and cancer on carcinogen mechanisms. And these are really considered in a very formal way through expert judgment to reach overall evaluations. And this slide really shows how this is done. So first, each of these lines of evidence is evaluated individually, and there are specific definitions for the terms that I've listed here: sufficient, limited, and for mechanistic evidence, strong. And this is how this is done. And IARC uses these terms, these famous groups: Group 1, Group 2A, Group 2B, and Group 3. And the terms carcinogenic, probably carcinogenic, possibly carcinogenic, and not classifiable. But other agencies may use different terms. They might call it carcinogenic to humans. Reasonably anticipated is used by the NTP, but the ideas are really the same. These are the things that are known. These are the things that are really suspected to be carcinogenic, and these are the things that are not. So over the history of the program, the IARC has evaluated over 1,000 agents, and there's a couple of things I want to emphasize here. So first of all, really about half of these have ended up in this bottom category, but IARC has also identified a number, really over 100 agents that are known to cause cancer in humans. And at the bottom of the slide, you can go to the website and peruse this in greater detail. But I thought it might be helpful to just emphasize how these Group 1 agents are identified and really what it takes to get to the top category. And it can be done in a number of ways, but the very, very large number were identified from human and predominantly observational studies. So we don't have usually clinical trials, but it's really investigations that are from the epidemiologist, that are sufficient in number and that indicate really that there is a causal relationship, that A causes B, that an agent causes cancer. And what are these agents? And I want to remind you here that IARC has a very broad remit. So it's not restricted, let's say for EPA, to environmental agents, or such as for FDA to agents that are drugs, but it's really a whole range of different types of things. And as you can see in this graphic, they include chemicals. This is ethylene oxide. This is benzene. You may have heard of those. Dioxins, but also occupations as a broad class. We know that. Fibers like asbestos. We can think about carcinogenic metals, a number of components of pollutants, including air pollution in general, tobacco in its many forms, including secondhand smoke, radiation, drugs, including immunosuppressive drugs, viruses. Now, viruses have been in the news a lot lately, and maybe people are tired of hearing about them, but some viruses are carcinogenic and cause cancer. And certainly for preventing cancer, antiviral strategies are very important. Vaccination is very important for things like HPV and also the hepatocellular viruses as well. So IARC reviewed all of these known human carcinogens about 10 years ago and made this very nice publication. And really this led to a number of advances that I'll come to later in terms of updating not only the cancer sites for individual carcinogens, but also identifying new carcinogens, and also integrate an opportunity to integrate data across what we know to build to the future. So this is another way to really that we can get from epidemiology that we can't get from really toxicology, which is to know what are the cancer sites. And I don't expect you to read all of this in detail here. You can go ahead and download it if you're interested to look more, but it does provide an opportunity for clinicians. This is one of the most popular downloads from the IARC's website. It's available in different languages. For any given cancer site, it identifies what are the known causes, and also in the greens, what are the opportunities for prevention. And some of these, of course, are going to include smoking cessation, enhanced screening for high-risk patients, and the like. And I'm just going to now, on the next slide, zero in a little bit on a couple of these cancer sites. And one is the lung. I think this is one where we know one of the best characterized. Doesn't mean that we know the cause of every lung cancer, but we've identified many causes that occur in occupational settings. Also things like outdoor air pollution, obviously tobacco smoking, but also opium consumption. And I just want to now contrast this with what we know about breast carcinogens, and just hearkening back to that first image that I showed you, and noting that breast cancer has become the most common cancer worldwide. We really have there's really a great deal that's not known about these causes. And why is that the case? And I published with Mary Schubauer-Berrigan, who's head of the Monographs program and who is an epidemiologist. We published a very nice invited perspective recently in environmental health perspectives, just highlighting some of the limitations. And it's important to remember that compared to the lung, well, many of these carcinogens were identified from occupational studies. These studies tend to be limited for looking at women's cancers, because the exposures to women may be very different. Women may be coming in and out of the workplace, and the study of them may be very much more limited. And so what we know about breast carcinogens really comes from dietary studies of medical interventions, including hormonal agents and also pharmaceuticals. So let me turn now to studies of cancer in experimental animals. I just want to emphasize that IARC is a little bit unusual in terms of how, in terms of the role of the epidemiology data, but for animal bioassays, I just want to say this is very typical. What you see here in terms of the predominance of the number of agents that have been identified really from animal bioassays alone. This is very typical for risk assessment processes around the world. And I think for many agencies, this is really can be considered the bread and butter of how we identify carcinogens, is really through these animal bioassays. They are done over the lifetime of the animal, and they provide a very solid evidence that people have a lot of faith in terms of identifying carcinogens. Now, you might say to yourself, well, but not all of these results can be relevant to humans. And of course, that is true. And some agents that are positive in these bioassays, through the process of considering all the different types of evidence, will end up in this last category, group 3. Now, I also want to take the opportunity here to emphasize that the words are important. And group 3 doesn't mean that there's no evidence. Actually, a lot of times for agents that are here, there is evidence, but it may be conflicting. It isn't certain. It's not mature enough to make a determination. And I think just as an example of an agent that is classified in group 3 is coffee. And so if you're drinking coffee right now, trying to stay awake while I'm talking, I would say go ahead. It doesn't cause cancer, so you're good to go. So let me turn now in the last aspects to the data on carcinogenic mechanisms. And to really set up this discussion, I wanted to harken back to a publication that I had the pleasure to participate in back in 2009. And what we did in this publication was to say, well, let's think about the number of chemicals that are in commerce and contrast that with the publicly available, these authoritative reviews. Now, more evaluations have been done by the IARC, by the US EPA, NTP, and this last one is the state of California. But you can see there is still a huge disparity between just the sheer numbers, including chemicals that may be produced in very, very large numbers, between what we know about them and just the sheer number. And so this sets up really a number of challenges with respect to identifying carcinogens. And the first is that if something hasn't been assessed, does it mean that there's no hazard? And secondly, well, given this huge number of things that haven't been assessed, well, how do we know which ones are of concern to communities, to scientists, and how do we select and prioritize those for assessment, and how could we improve that process? And finally, can mechanistic evidence help to really fill this gap and bridge across from what's unknown to what is known, and in distinguishing those that have no hazard to those that do. And why do I want to talk about the mechanistic data? Well, first of all, it is increasing in volume, it is increasing in diversity, and it has several opportunities. And mechanistic data really can range. You can do mechanistic studies in humans, you can do them in mice, and these abbreviations up here is really about throughput. This is low throughput, medium throughput, high throughput, and ultra high throughput, where you can literally just sit at your computer. Well, I can't, but other people who are gifted in this can do modeling exercises. In these types of experiments, you can also do cellular experiments. High throughput assays have been developed, including by the US government, but you can also think about alternative species. A lot of progress with zebrafish and other organisms. And these different approaches really range and have different strengths and weaknesses. And this is one of the issues that was addressed in the article I had mentioned earlier from EHP, that really there are different opportunities that remain untapped. But what we really need to know is what's relevant to our question. Our question is, what causes cancer? And some of these are going to provide information that's interesting, but may not answer that question. And so this was a conundrum, I think, faced that the field of toxicology has really faced and wrestled with for a long time. And so through this paper that I mentioned in 2009, what we thought to do was, well, could we make a list of possible mechanisms of the human carcinogens, and then somehow use that information as a lens to agents where we're not sure if they are carcinogenic or not. And you can see this list of mechanisms covers a lot of different things, and aflatoxin B1, which is a known carcinogen. All of them are. Arsenic, asbestos, benzene, and diethylstilbestrol. They have different of these mechanisms and really different properties. And so this exercise was really informative to the thinking but it was still unclear what should we do about this and how to go forward. And so through some workshops that were actually conducted at the IARC, this concept of could we take these human carcinogens that I showed you before and use them to provide new insights. And this concept of the key characteristics of carcinogen was really brought to the fore by Martin Smith, who's at UC Berkeley. And the idea is that these carcinogens have properties, and you can think about them as the top 10 properties. And I would challenge cancer scientists and others, if you made a list of the top 10 properties of human carcinogens, it might look very similar to this list itself. And we have agents like ethylene oxide that really interact with DNA. We can think about our fibers, which cause chronic inflammation in the lung. We know that, reactive oxygen species. We have our drugs that are immunosuppressive. Well, that's what they actually do. And that turns out to be how they cause cancer. We have our hormonal agents that are obviously modulating receptor-mediated effects, the viruses which are causing immortalization, and also many of these other properties from which you get from one cell to the other. So we've published quite a bit on this. We have a website on the key characteristics that I would encourage you to access and to get involved in some of the ongoing projects. So, so how can this be used in considering carcinogens? And as I mentioned before, it's important not only to know what is relevant, but what isn't. And we know that when we have evidence that that for instance, if you have that sufficient evidence of cancer in experimental animals, but you have strong evidence, this is a mechanism not relevant to humans, you could actually move it down to not classifiable. But actually the data from other mechanisms that are relevant, that are considered what we call relevant to the key characteristics, can be used alone and in combination with other data streams to really identify carcinogens and to actually classify them into this top category when combined with sufficient evidence of cancer in experimental animals. So I'd like to close with this publication from the US National Academies of Sciences. You can download it. It's actually one of our most popular download. A really, really, really outstanding committee chaired by Jonathan Samet. And the committee really looked at some of the different approaches that are available in terms of mechanistic data. And they considered that this approach based on the key characteristics had some promise because it avoids a narrow focus on a specific pathway and really provides for a broad and holistic consideration of mechanistic evidence. And they actually encourage the development of cases for other hazards. And this has now happened for things like cardiovascular toxicants, reproductive toxicants, and a range of different scientists from different disciplines have found this to be useful. So I'd like to close by thanking the many, many scientists from around the world who have contributed through their volunteer work at the Academies, at the IARC. I'd like to also thank the IARC Monograph staff. And especially I'd like to thank you for your attention. And I'm very much looking forward to the discussion and to the presentations that are forthcoming in this session.
So Kate, if you'd like to look at the chat and answer a couple of questions. I don't know if Ruth Ann would like to choose a couple questions for you to focus on.
Check the Q&A, Kate, and you can see what you have time to answer.
Yeah, sure. So I can let me take a couple of easy ones. So the group three the group three, everything in group three has actually been evaluated. So I think that's an easy one there. What else? I don't know, Ruth Ann, if you wanted to just bring one forward. I think there's a lot of questions also about animal testing and moving away from that. And I think this has really, I think in Europe, there's various laws that have really moved away from whole animal testing to more in vitro methods or new approach methods. And this has really been this has been a motivator for thinking ahead to how we can really best use mechanistic data to make those kinds of confident decisions. As I said, epidemiology is wonderful, and it can provide more confidence in whether things cause cancer, but it's also not a way in which we can predict the future. Epidemiology is really looking at what already happened. And unfortunately, those people already had cancers that can be examined and studied. Ideally, we would not have cancer in humans. We would be able to prevent that by preventing exposures that we know or suspect to be carcinogenic. So it's been a great motivator, and I think there's a lot of interest around the world in how we move forward with these new approach methods.
Kate, there's a question actually from Margaret Kripke. I'm wondering if you could answer why there isn't a category for non-carcinogenic.
Yeah, Margaret, that is a great question. So actually in the last, before this latest update to the IR preamble, there was a group 4, which was not carcinogenic. And I think that there was only one agent in it, and it kind of created a lot of confusion because it's really based on what's been studied, which in some cases doesn't cover all scenarios. And so you're drawing a very narrow conclusion. And the advisory group that was convened to update the preamble, it opined that it would really simplify matters if we just combined groups 3 and 4 and put everything in this not classifiable. So, but a lot of other agencies do use the not carcinogenic classification. I think it can be unfortunate if you put something in there and later on you find with new data that it is, and that maybe creates more confusion than not. So I think there's different approaches to that. So another one, the status of the Ames test. You know, the Ames test, it has been around for a very long time, and it has an amazing predictive potential. It can predict the results of rodent bioassays about 85% of the time, which is pretty good, I would say. And I think that a lot of scientists still rely upon it. I think the most batteries would consider the Ames with other types of assays. It kind of evaluates one aspect of genotoxicity, and of course there are others, but I think over time it is really held up as a very valuable tool for understanding potential hazards.
Thank you so much, Kate. That was a fascinating and informative talk. Just to keep us on time, I'm going to suggest that one or more of us can answer some of the questions in the chat and move forward, because there's some really great questions there, and I would like it if they can be answered. So thank you again for your talk. And I'd like to now introduce Mary Beth Terry. Mary Beth is a professor of epidemiology and environmental health sciences at the Mailman School of Public Health at Columbia University. She's also associate director for population science and community outreach at their Irving Comprehensive Cancer Center. She's focused her research over the last 2 decades on understanding the role of environmental and lifestyle factors on modifying cancer risk across the life course and in higher risk populations. She leads large cancer cohorts, including the Breast Cancer Family Registry and the New York Breast Cancer Research in the Environmental Program Adolescent Cohort funded by NCI and NIEHS, and was recently funded to head a multi-institutional New York City region Center to Reduce Health Disparities in Multiple Chronic Diseases, also funded by NIH. So Dr. Terry is going to speak on precision prevention, gene by environment interactions, PAHs, and the breast cancer family cohort. Mary Beth.
Wonderful. Thank you so much. I'm just going to share my screen then. And I wanted to thank the organizers of this conference and also to thank each and every one of you for participating today. It's a privilege to be here. I was asked to focus my comments today on what we have learned about the causes of cancer and specifically how cancer genomics have provided context for understanding the role of the environment in cancer causation. In doing so, I will argue that the human evidence is much more consistent when we consider underlying susceptibility to cancer, and I will give a specific example with my own research in breast cancer and polycyclic aromatic hydrocarbons. To frame the talk, I would like to contrast and start by recognizing that the approaches that we take for population health and disease prevention can be very different than the approaches we take for discovery. For example, although the entire population should benefit from policies and interventions that are effective, and we should always evaluate what happens when the population shifts from a harmful environment, as in A, to a less harmful environment, as in B. But discovery for cancer genes, as well as cancer environmental factors, may benefit from studying those who are most susceptible, as is shown on the bottom right. Over the past half century, many causes of cancer have been discovered, including some of the factors that I list on the left. It is important to note that cancer control of many of these factors has happened through policies and regulatory actions, as well as individual level changes, and that we need both policies and regulations, as well as individual level interventions to control cancer. For example, since moving to New York City in the 90s, I've seen the smoking rates cut in half through citywide smoking bans and other such policies. This is not to say that individual level tobacco cessation programs are not extremely valuable. They are, but rather to emphasize again that cancer control needs both policies and regulatory action, and success that we have in influencing policymakers, particularly you as healthcare providers, can be very influential, both in terms of transforming public health through regulation and new policies, but as well as individual health, as you do with your patients. Over the last couple decades, there's also been major gene discoveries. There are now known to be hundreds of cancer genes that have been discovered, many of which alter the same mechanisms that you saw in the first talk, and relate, for example, to how we control damage that we may have from many different carcinogens. It's important to know, unlike some other genes that cause different diseases, that there's no single cancer gene that's 100% penetrant, meaning that even if you're born with one of the alterations in these cancer genes, that there is not 100% guarantee that you will get cancer in your lifetime. As an aside, we should also observe that the causes of cancer, whether they're on the left or the right side of this slide, have been, as was mentioned by the previous speaker, discovered through observational non-randomized studies. So observational studies, even though they may have additional biases than randomized clinical trials, really they are what we rely on for determining causality, and they have transformed population health. Thus, for all of the cancers, it's not either or, but both and. For example, cancer genes that have been discovered in families such as BRCA1 for breast cancer is just as important for women without a family history. Many cancer genes we now know can be altered throughout the life course, and therefore susceptibility to cancers can be changed both through endogenous and exogenous factors throughout life. Environmental factors may be even more important for those at higher susceptibility and may in fact influence susceptibility as well. Here I illustrate the importance of modification of risk with the example of physical activity in breast cancer. On the left, the relative risk reduction from physical activity is similar, irrespective of an individual's breast cancer risk, which we predicted based on a woman's known genes in breast cancer susceptibility genes, as well as her family history. So the similarity on a relative risk scale means that all women would benefit from reducing physical inactivity. But on an absolute scale, it has a much bigger impact for those that are already at higher risk. So again, it's not either or, but both. And yet what happens when risk is communicated and attributed? First, we continue to simplify the picture by combining all cancers together, even though we know that the causes of different cancers is very diverse. And we try to quantify the impact of single risk factors, even though individuals live our lives exposed to many factors simultaneously. For example, you can see on the right here, a pie where there's an attribution for family history. Again, as was mentioned, family history alone cannot cause cancer. It needs to interact with environmental factors. The clearest example of this is twin studies, which have supported that even if you share 100% of the genes with your sister, in twin studies, this would be referred to as a monozygotic twin, and she has breast cancer, your cumulative lifetime risk of breast cancer is 30%, meaning there's a 70% chance of not getting breast cancer, even if you have 100% of the same genes. Here on the right now, I've contrasted the current updated American Association for Cancer Research figure with the one on the left from 5 years ago. What's missing? So if we look a little closer, we can see that occupational exposures, environmental exposures, and reproductive factors are missing, even though the data to create these two graphs has not changed over the last 5 years. Why might this be? We don't know, but we could speculate that the emphasis on the right-hand side is to focus on things that may be more modifiable at the individual level. But again, as we saw earlier, that the causes of cancer and cancer control requires both individual level modification as well as societal and policy and regulation modification. Individual risk factor modification may be easier to apply, particularly for busy health care providers such as yourselves, who have so many demands on your time. Yet, you can understand that valid questions remain. A key challenge in understanding inference really comes to the fact that, again, when we think about attribution, cancer is many, many different types of cancer. And WHO, for example, attributes 1/4 of all lung cancers and cancers of the trachea, as well as bronchus, to environmental exposures, as the previous speaker mentioned. And so when we talk about the attribution and people then may think that the environment doesn't matter as much as some of the other risk factors, we need to keep in mind all of these considerations. Further, given that exposures, people are exposed to many different things at the same time, what we again attribute at a population level sometimes is easier to do for things that are easier to measure, such as cigarette smoking. These are key considerations when we're trying to understand what are the big drivers of cancer risk. So the cancer trends themselves have differed dramatically over the last 40 years. And again, a lot of this is attributed to improved screening, but certainly public health and reductions in smoking, which have been very dramatic over the last half a century. What you can see here is that most cancer occurs in individuals who are older. And so the yellow line for women on the left and men on the right are adults over 70. And you can see most cancer occurs in older individuals, and the trends have been going down, but only really going down in older individuals. In fact, even though cancer is rarer in younger individuals, the trends that have been occurring over the last few decades have been very dramatic. And particularly if you look at the increased trends in adults under 55 and women under 40 is really startling. So the increasing incidence, again, is not driven by genes, right? But has to be driven by environmental changes and environmental exposures interacting with underlying susceptibility. So I'd like to just focus on what I know more of, which is breast cancer. And again, globally, cancer incidence has been changing dramatically. And that is true for breast cancer, what you can see right here, which is the #1 cancer in women around the world, which was not true just a couple decades ago. In fact, in countries like India, where cervical cancer was much higher than breast cancer has now surpassed it. And what we've also learned over the last 30 years is, and this is coming from both human studies as well as animal studies, is that the breast tissue itself is much more susceptible during key windows of susceptibility, sometimes referred to as WOS. And these windows of susceptibility are times when the breast tissue is changing in form and function, and therefore can be more susceptible to carcinogens. And those include the prenatal period, the pubertal window when breast budding occurs, during parity and lactation, as well as the menopausal period. And yet, most of the studies of the environment in breast cancer, including studies of dioxin, organochlorines, polycyclic aromatic hydrocarbons or PAHs, air pollution, and other endocrine disruptors, you can see here on the left. This was a review article done by Drs. Rogers, Udesky, Rudell, and Brody. And it was a very comprehensive review article looking at all the publications within a 10-year period. And there were a lot of publications, but yet only a minority of those publications actually specifically addressed the question of carcinogens during those windows of susceptibility. And in fact, most of those studies were all primarily animal studies with a few human studies. So again, there's a large base of evidence from human studies, but most of those studies are from human epidemiological studies that are outside a window of susceptibility. So if we take a closer look, though, at those windows of susceptibility, and we look at just those human studies that have measured these certain environmental exposures during those windows, you see much greater consistency. So, for example, you can see increases from organochlorine exposures, from prenatal exposures, and exposures during puberty. You can also see, for example, metals exposure during a window of menopausal transition, and also organochlorines, PCBs specifically, during the pregnancy window. So again, the consistency comes when the question is being asked, which is what we know biologically to be the windows where breast tissue is changing. We had published, for example, a study using a birth cohort from California and looking specifically at whether or not windows, for example, exposure before age 3 or in utero or around the pubertal window, how that related to cancer then later on. And you can see here, again, this was using an older birth cohort, which was really addressing what a lot of epidemiological studies can't do, which is measurements specific to a window of susceptibility. You can see that there's a larger association twofold that becomes even much higher when looking at early onset breast cancer. So again, when we can look more at susceptibility, the evidence is much more consistent. We conducted a similar systematic review, but asking kind of a different type of question about susceptibility. And specifically what we were interested in is whether or not the evidence base for environmental chemicals was stronger when you looked at individuals who were at greater susceptibility. So keep in mind, many of those epidemiological studies, not only did they not have specific windows of susceptibility when they were conducted using the environmental exposures, but they also primarily contained a lot of individuals who were of older ages. And so the underlying susceptibility based on underlying genetics would be very different later in life compared to looking more at early onset cancers. So we looked at 3 types of studies. The first type was enriched studies based on individuals' family history. The second type was specific to early onset cancer in premenopausal women, just like what I showed you in the slide before. And then the third was really looking at gene environment interactions. What you can see from this systematic review, again, we classified the chemicals similar to the other review article. What you can see is most of the studies, again, in green refers to more data being available, in blue, less data, and all the zeros mean there's absolutely no data. What you can see is that most of the studies were of the type of gene environment interactions. And so again, we can ask ourselves, how consistent is the evidence? And what you can see is that when we look specifically at people who we know to be at higher risk for breast cancer, that the evidence is much more consistent. Over 70% of the type 3 or gene environment interactions were positive in subgroups of women with greater genetic susceptibility. Over 70% of the publications from the enriched cohorts, type 1 or type 2, were also positive. So even though you can think about a much broader literature, again, just like the windows of susceptibility, it's much rarer to have studies that specifically look at people with higher genetic susceptibility. And again, these include genetic variants in carcinogen metabolism, DNA repair, oxidative stress, and cellular apoptosis, as well as tumor suppressor genes. So again, if we're thinking about if we're thinking about what to consider and how to review the evidence, most cancer does happen in older adults. But again, what we want to understand is to measure the signal when we're including the bulk of cancers from older adults, we have a very heterogeneous group, which includes adults where their cancers may be related to endogenous processes related to aging. Whereas when we look at early onset cancers in life, we have a much greater homogeneity when it comes to risk in thinking about that. So we conducted a study of an enriched cohort, which also it's important to mention that individuals with a family history have a very different absolute risk. So what you can see here on the left highlighted in blue are all of these individuals come from families with a breast cancer family history, but there's many thousands of women based on their family pedigrees, which are actually at lower than average risk. So if the average risk is 1 in 8 for breast cancer in women's lifetime, you can see even women with a family history, again, based on what's known about their family pedigree as well as their known genes, may have even a lower risk. And then also within these kind of enriched cohorts, there's many that have higher risk based on known breast cancer susceptibility genes. So these enriched cohorts, unlike population-based cohorts, have a wide range of absolute risk as predicted here and shown on the x-axis. And so that gives us better statistical power to examine possible causes of cancer. And so that design we've now used with a few different risk factors and understanding environmental causes. So I'll give you a few examples then from our research looking at polycyclic aromatic hydrocarbons. And polycyclic aromatic hydrocarbons, many people are exposed to daily, again, through air pollution, through tobacco smoking, through fires, through also just the way we cook our meat. And the Long Island breast cancer study, which was really the first of its kind to examine polycyclic aromatic hydrocarbon in a very large population-based study, I had the privilege of working on that as a doctoral student, and using a biomarker approach, you could see there was a modest association with PAHs overall, and that association got stronger when you were able to subgroup it based on someone's underlying genetic risk. And again, these were the early days where you kind of looked at one genetic variant at a time. What we can do now, however, is really, again, combine all of what we know about someone's underlying susceptibility, including now a polygenic risk score, which allows for looking at over 300 different genetic variants, as well as intermediate and high penetrant genes. And we can do that on the x-axis here. This is a woman's absolute predicted risk. And then what's in pink is, and the other colors is based on her blood measurement of protein addicts to polycyclic aromatic hydrocarbons. You can see for women who are heavily exposed and also at higher risk based on their underlying susceptibility, very, very large odds ratios. Again, in this zone, epidemiologic studies then are much easier to say that this is not because of bias, because these associations are so large that it's unlikely that bias could remove those associations. So you can see for highly exposed women that these associations were quite strong. And this kind of method of study gives you the ability to detect a signal that would be harder to do and would be smaller when you have a very mixed group of people with different underlying genetics. So in thinking about then where to go with this, as a healthcare provider, you provide an essential part in communicating risk. In the case of breast cancer in the environment, if we were to merely count up the studies, the evidence would look inconsistent. I'd be the first person to say that because there's hundreds of studies. However, as I showed you today, that most of those studies were not conducted within windows of susceptibility. And also most of them did not account for someone's underlying genetic susceptibility. When we do look at the relevant windows of breast tissue susceptibility as well as genetic susceptibility, the evidence is much more consistent, and the conclusion that you would reach is very different. So how can we respond to questions related to whether environmental factors may cause breast cancer? To be truthful and describe where certainty exists and where it does not take away hope. It reinforces trust. Words matter. In conclusion, we have come a long way from the simplified pie, and we now know that cancers are caused from interaction of the environment with underlying susceptibility. It's not either or, but both and. And of course, both environment and susceptibility change over the life course. We can use enriched cohorts to find signals of cancer causes, just like we do with gene discovery, and just like we have done with many of the hundreds of cancer genes that have been discovered. But these findings then need to be applied to the population as a whole so that everybody can benefit. Cancer control will continue to require both population-wide policies and regulations, as well as individual level interventions. As healthcare providers, you play a pivotal role in both. Thank you.
Thank you so much, Dr. Terry. That was a really interesting talk. I have a couple questions. You can feel free to look through the Q&A and pick what you want, but I was going to help you out by asking a couple. There are a couple questions about whether aging, alcohol, and obesity would also increase susceptibility to environmental exposures.
Great. Maybe I'll take the alcohol and obesity. So the obesity part, there are a number of the environmental exposure and a number of different endocrine disruptors specifically have been shown to be obesogenic. And also there's been a lot of data to support that we store environmental chemicals within adipose tissue. So yes, I would agree that those interact. I'm less clear on the alcohol interactions, although alcohol itself has been shown to interact with a number of different other factors, for example, folate, and that's been shown for a few different cancers, including colorectal cancer. So yes, all of these things do have different interactions that, but need to be considered simultaneously.
Thank you. I don't know if we have time for one more question. There's a question about the point that you made about measuring exposures and doing attributable risk calculations. So how is it that smoking, or something that's easy to measure like smoking, is affects calculating attributable risks when you have other exposures that might cause cancer but are harder to measure?
Right. I mean, in essence, it's a great question. The individual has honed in on the big challenge, right? So what's in a statistical model, what explains the variance gets the most credit, in brief. And so it is much easier to measure things like smoking. We can measure smoking pretty validly by questionnaire and pretty validly retrospectively as well. Whereas, we know that environmental chemicals are much harder until, now more recently with big data, we can link to other sources for that information, and we can collect information on, for example, residential history to get kind of exposures from the past, but they are harder to measure, they're expensive to measure, and there haven't been as much studies as when you can measure things simply by questionnaire.
Thank you.
Thank you, Mary Beth. That was great. And I see that Kate Guyton has been answering some of the questions in the chat. Mary Beth, if you could do the same, that would be really helpful. This is some wonderful questions there. Really appreciate your great talks. Thank you. Next we have what I think will be a very compelling and interesting case study and panel discussion led by Dick Clapp, who will be introducing the Wilmington Childhood Cancer Cluster Study and moderating this panel with some stakeholders. So Dr. Clapp is an epidemiologist with more than 40 years experience in public health practice, teaching and consulting. He's Emeritus Professor of Environmental Health at Boston University School of Public Health. He served as the first director of the Massachusetts Cancer Registry and has been involved in many investigations of environmental and occupational cancers, including childhood cancers in Woburn and Toms River, New Jersey, and for workers at IBM and Marines at Camp Lejeune. So I'd like to start, though, with a very brief video that gives a great introduction about Dr. Clapp. So if we could start that, and then we'll have the rest of the panel.
Actually, I don't think we have the video, so I think we should.
I'm sorry. Then we will not do the video. We will just go to Dr. Clapp. Welcome. Thank you, and take it away.
Thanks very much. Thanks for inviting me to be a part of this. This is the first two presentations were incredibly rich. What a treat to hear both of those. And it's really, state-of-the-art, up-to-date work. So it's a pleasure to be part of this. I thought I would first give a little background about the cancer registry that was so critical in the Wilmington study. Somehow my video just seems to have gone off, and I am trying to, there it is. I was hired in 1980 at the Massachusetts Department of Public Health to establish a statewide cancer registry. And it was because of Woburn. Dr. Rebbeck mentioned the childhood cancer cluster, childhood leukemia cluster in Woburn. Once the Woburn cluster was identified, it was really by citizens and parents of children who had been diagnosed with leukemia, but there was no statewide system for keeping track of leukemia in children or cancer of any type in other people, other than death certificates, those who had died of cancer. So the state legislature passed in 1980 a law mandating reporting of cancer diagnoses in Massachusetts residents as a reportable disease. So I was hired to set up the mechanism for doing that. And it was really because of Woburn. And as a result, we were trying to make sure that we identified hotspots or places where there were excess cancers of some type, including in children, around the state. So we set up a system to make sure that that kind of information was available where people lived and in the adults where they worked, and some other factors that would determine the likelihood of there being an increased risk of cancer in their community. And so that's what happened in the 1980s. I was director from 1980 to 1989, and the childhood leukemia cluster in Woburn was studied in more detail by collecting information from the cancer registry and then the previous cases that had been identified by the family members, and going back and asked where they lived, where they got their drinking water, whether there are other risk factors, so-called case control study, the cases of leukemia, or the parents actually that were interviewed of the cases of leukemia, and then parents of controls who were either from the same daycare center, same school, or the same community. And then there was a comparison of whether the cases were more likely exposed to something in their environment. And the results of that study, which was published in 1986, suggested that if you were a resident of a part of Woburn, Massachusetts that had contaminated drinking water, contaminated with two solvents, trichloroethylene and perchloroethylene particularly, your increased risk was due to that residential exposure. But even further, if you were exposed while the mother was pregnant, if the child was exposed in utero, in other words, then the risk was even much higher. It was in the level that Dr. Terry was referring to as no question. It was 8-fold increased risk of leukemia if you were exposed while in the uterus. So that led to a whole series of similar studies. Dr. Rebbeck just mentioned Toms River, New Jersey, where basically the same method was used to investigate a childhood cancer cluster in Toms River, New Jersey. The key part of this was the water model, where they could look at the known contaminants that were in the drinking water by well water region, and then they could estimate hydrologically how much of that got to the homes of the people in the study. In Woburn, the water model was developed by a person who at the time was at UMass Amherst. Peter Murphy was his name. He actually served as a consultant to the Centers for Disease Control, the ATSDR agency in Atlanta, which did the Toms River, New Jersey study along with the State Health Department. And the water modeling got more and more sophisticated, where you could look at things like prenatal exposure. And sure enough, in Toms River, they also found that increased risk of childhood leukemia in those children who were exposed in utero before they were born. And there have been subsequent studies, including the Camp Lejeune study. There was a subgroup of children born on Camp Lejeune, or born while their parents resided on Camp Lejeune. And that showed similar, it was a small study, so that it wasn't as statistically robust, but nevertheless, it indicated the same thing: increased risk of leukemia and lymphoma, actually, in children to Marine families that were born while their parents were residing on Camp Lejeune. So all of this is background to the Wilmington situation. Wilmington, of course, is a city, a small city in Massachusetts that is covered by the state cancer registry reporting law. And so there was data showing up in the 1990s when parents actually became concerned, parents of a particular neighborhood, because of known contamination from a chemical company that had been identified in drinking water. And there was a suspicious increase in childhood leukemia and childhood cancer. And so that was, again, that sort of red flag that was set up by the reporting system, showing that there was, in fact, an increased incidence of childhood cancer in Wilmington. And then a subsequent study, a follow-up, what's called a case control study, interviewing cases and controls, cases of leukemia, and then patients who had did not have that diagnosis, but lived in the same general area. And the water model was used. In fact, it was a consultant from, two, one from Tufts University and another from the Centers for Disease Control in Atlanta, that verified a water model of the distribution of contaminated water within Wilmington. that led to a final report that was issued a year ago, actually, a study that showed once again that childhood leukemia in Wilmington seemed to be related to exposure to contaminated drinking water from chemical contaminants. I should mention that trichloroethylene was found in all three of the previous examples that I just mentioned in the drinking water. So that was one of the contaminants in Wilmington, along with a nitrosamine-related chemical called NDMA. So it was a mixed exposure to both trichloroethylene and NDMA, so it wasn't possible in the Massachusetts Wilmington study to identify a particular chemical that caused this increased risk. But once again, prenatal exposure was where the greatest risk appeared. If you were a child that was later diagnosed with leukemia, you were at particularly increased risk if your mother had been exposed during pregnancy. So all of that is a way of saying there was an important study that was recently released in Massachusetts that again documented an association between childhood cancer and chemical contaminants of drinking water, and that we have three panelists I'll introduce now, and we're going to do a kind of a lightning round or a speed dating of each of the three panelists, and then have time for questions and answers afterwards. So the first panelist will be, and I'll just list them now in order, but I'll then ask Brian to start out. Brian Delascio is a Wilmington cancer survivor. He himself was diagnosed when he was a child and has become a patient advocate subsequently. Next, we'll hear from Kathleen Barry, who is a Wilmington resident, but also served in the recent study as a community liaison between the Department of Public Health in Massachusetts and the local community. And last, Mark Nascarella, who is the chief toxicologist and at the Massachusetts Bureau of Environmental Health. And he will talk about a survey of clinicians, a survey that he did, he and his colleagues at Mass Department of Public Health did, of their clinicians' experience with environmental exposure. So as I say, we'll have about 5 or 7 minutes presentations by each of these three, and then we'll open it up for Q&A from the audience. So Brian, please take it away.
Thank you, Richard. Thank you for inviting me to this. I think this is an important topic for a lot of people to be aware of and to become advocates, because I think it's something that affects so many lives everywhere, and it's going to continue to affect us until we start mitigating some of the dangers here. So just to give you a little bit of a history on my medical past, I was diagnosed in '91 with Hodgkin's lymphoma, and I want to append something Richard said about leukemias, but I think it was blood-borne cancers. So there's lymphoma, there's leukemia, and a few other cancers. No problem. So I was in the Hodgkin's lymphoma category. So I was diagnosed in '91. I had my initial staging operation, splenectomy. I was found to be stage 2B, which is fairly early in the overall arc of stages. And it turned out that the cancer was the wall of my lung, and I had presented above my clavicle with an enlarged lymph node. Upon that diagnosis, I did 10 weeks of radiation, 5 above and below the diaphragm. I got initial, I completed that, and I relapsed immediately. Then it had spread to my small intestine, my kidney, my liver. And it seemed like it was behaving more like non-Hodgkin's lymphoma, which tends to be more aggressive, but there was never any conclusive evidence that my cancer was non-Hodgkin's. So I've just always been labeled as a Hodgkin's patient. After that, I went through 10 months of chemotherapy. At that point, they had taken care of everything but what was in my liver. And at that point, there were conversations that started around having a bone marrow transplant. So my bone marrow was clean, so I was able to do an autologous bone marrow transplant. That was after another set of chemo to reduce the liver tumor as much as possible. I was in remission after that for the Hodgkin's, and ongoing challenges with that are immunocompromised state because I don't have a spleen. Occasional bouts with shingles here and there. But in general, was in pretty good health. Then in 2005, I was diagnosed with a secondary cancer, colon cancer. I went through chemotherapy for that, was in remission in 2006 for that. And then the most recent medical challenge I've had is I actually had a heart attack, and I had heart surgery, and I had a valve replacement done. And it turned out that some of the heart issues came up from when I had the radiation as a child. They blocked things with the radiation back then, but it wasn't as precise as it is now. They can really target the radiation in very specific places. And they said that one of the risks back then was thickening of the arteries, which actually cut off the blood from my mitral valve and caused problems with my mitral valve, which then prolapsed, and I had a heart attack because of that. What's interesting is that I operated for about 2 weeks in heart failure and didn't know it. I was driving back and forth to work. So I found that out. I was immediately hospitalized, had that surgery, and I've been okay ever since, outside of normal, ongoing issues. So that's kind of my medical history with that. So go ahead, Richard. Did you have another question?
Well, one of the questions that comes up is, what's your understanding of the connection between the exposures that you had from drinking water and the cancer that you got diagnosed with?
So my understanding is that it's prenatal in my situation. So there were contaminants in the wells when my mother was pregnant with me. Not too long after she was pregnant with me, the wells, I think, were shut down for a bit in Wilmington because there were other things that had come up that they had found out about. They shut the wells down to do the mitigation for those issues. So there was a gap in those chemicals being, introduced into the water supply. Then the wells were reopened again, which then reintroduced some of the chemicals they weren't aware of and didn't know about. And then that was kind of the ongoing thing until much later. The wells were closed again. So there was sort of a gap in the exposures. But I was one of the earlier cases that I presented in the early 90s. But I was 15 when I was diagnosed. So my mother was pregnant with me in the mid 70s. So that's my understanding of the exposure.
And how is this your experience shaped your views about environmental chemical exposures and their ability to cause cancer?
So I think this is more about the response to it, I think, which is, community engagement and action is critical to help bring about change. Having a coordinator like Kathy Barry, who you'll talk to in a minute, she understands really how to navigate the public health and some of the political arenas that you end up getting yourself into with these kinds of situations, because there's obviously local politicians and things that don't really like to have these issues come up for their town or their city that they represent, because obviously there's a lot of negative connotations around this type of thing. So there's a lot of usually political sorts of things that play there slightly under the surface that may not be obvious to everybody. The initial study, once it was completed, we're talking 20 some odd years ago now, it was definitely an exercise in patience for the families. But we were all in agreement that, to make sure that the data that we got out of this was solid, that it was vetted, that it was, there couldn't be holes poked in it, because then the entire effort would have sort of been wasted in a way, because we wouldn't have been able to get solid data that, was backed up and vetted by folks that know what they're talking about. And that led to the study, which made, I believe, the first connection between NDMA and cancer. So that made it worth the wait, although it was, a couple of decades. There's still a lot that we don't know about chemicals in environmental contamination, but we're finding out more and more all the time. There are multitudes of chemicals in Wilmington that couldn't even be identified. That's how bad some of the contamination was from that site. And I really do feel that the goal of, having community cancer cluster investigations is to, get that solid evidence to then help drive future public policy, trying to get, the right limits put in place for what's acceptable, what can be in a water supply before it's considered a danger, as well as other mitigating factors, making sure that chemicals are vetted and tested, and, all of this stuff sort of spills out, I think, from getting that solid evidence that can link these chemicals to cancer. And then the other piece that I learned was that public-private partnerships can be a really powerful tool. There was a number of partnerships done between the state and some educational institutions that really helped us get our hands on some of this data and the modeling that we otherwise wouldn't have had access to. And that helped tremendously. So taking advantage of our incredible educational institutions here in the US, and especially in Boston, we're very fortunate, but I know we worked with some other institutions outside of Massachusetts, but partnering with those institutions as well can really help bring about a lot of helpful data that can help drive that policy forward.
That's great, Brian. Thank you for that. And we'll have time for more questions as we finish with the other two presentations. So next, Kathy Barry, please take it away.
Thank you, Dr. Clapp. I really appreciate this opportunity to talk to the audience and the participants, how important community advocacy is in driving a potential concern. Back in the 1999 and 2000, actually, with two, just like Woburn, two parents, two mothers, who recognized an increasing number of childhood cancers in a particular neighborhood. They went to the public schools, they talked to the nurses. And then, well, we were unfortunately under the confines, I should say, of the HIPAA laws and rules, where they couldn't share with us anything with regards to the numbers of cancers that were arising in the community. So those parents went to the Board of Health. They wanted the Department of Public Health to investigate. The doors were closed there. They went to their state rep, who also lived in the area, which was fortunate. And he, in fact, petitioned the Department of Public Health to do that study, to do the study that eventually expanded through the years. I was drawn in by a Woburn mother who was a friend of mine, and she said, you know, there's a problem in Wilmington. I said, no, I didn't know there's. I only knew of a contentious meeting at the Board of Health. And so she drew me in, and it became evident that it is so hard for parents to deal with their children's cancers and also have to fight City Hall for the just basic knowledge and truth. So the community advocacy and the role that I played as a liaison to the Department of Public Health was to help alleviate that stress and anxiety that they knew that those few people at the time knew that there were good heads coming together, looking at the situation. It took many years. It took many years of fact finding. We took many, we broke up into community groups. We looked at how we could work with the state and federal agencies to find out the answers that we were striving for. And our most, with as far as the cancer cluster was concerned, is we developed a very strong relationship with the Department of Public Health. The community assisted in the questionnaire, in the interview processes. The community worked with them very, very closely, and they had developed a strong partnership. And we knew what was going on all along. They kept us involved, and we were able then to, over time, get the group of families together through putting an ad in a local paper and asking them to come to a meeting at the local library so that we could discuss on how we could find out who was involved and the truth for those that were striving to know the answers. And so it is a long, arduous process, but very well worth it in the long run.
You mentioned that the local health department, at least initially, was, I'd say, dismissive or not cooperating. How could that change? Or what did you learn from your experience with this in Wilmington that might affect how other communities, or even the medical community in other communities, physicians, nurses, healthcare providers?
Yes, that was very, very distressing for all of us, thinking that it was a local Board of Health. We could actually almost see it in our select board and people that were protective of values, home values and the like, but our Board of Health is there to help us with health concerns. I guess to help, articulate now, well, hopefully this town will believe that now that they were somewhat dismissive, always even up to 2 years ago, very, very dismissive of whether there would be a connection made. And I think it was a matter of, they wished it not true. They wish it would go away.
Yeah.
I think that, those lessons are learned, and I think the best thing is for all of, when there is a cancer cluster and concern, look what happened with Woburn to us. We can then become advocates in those communities and detail our experiences and try to break down those walls, because eventually it becomes a very positive finding case. And now you recognize how much work has been done to identify, to protect further health issues in the town.
Well, good. Thank you, Kathy, for that. And I think that's a good way to segue into Mark's presentation about what do physicians know about all of this, or what could be learned from the survey that he just did. Can you tell us a little about that, Mark?
Sure, Dick, I'd be happy to. And to sort of set the table, in Massachusetts, we have over 44,000 hazardous waste sites, although there's probably about 30 or so Superfund sites like Woburn and like Wilmington. So not all of them are the same scope as the sites we've just been discussing, but they do raise a fair amount of concerns with residents in the Commonwealth. And oftentimes those questions are brought to the physicians and nurses and primary care providers that individuals see. So through a cooperative agreement with the Federal Health Department and the Agency for Toxic Substances and Disease Registry, we conducted a survey of primary care clinicians in Massachusetts to understand what is the baseline environmental health literacy and capacity of primary care providers to have that conversation with their patients about environmental health. We sent out a survey to actively practicing clinicians in family medicine, internal medicine, and reproductive health. The survey included questions really in 4 different domains: health assessment and counseling, educational preferences for information, patient education, and the respondent, the clinician, as well as the characteristics of the patients they serve. So we had over 680 primary care providers in Massachusetts complete the survey, or at least start the survey, and 500 total completes of the survey. So it was a convenient sample. We have some sort of an idea of primary care capacity and capability, but certainly not every healthcare provider in the state. The participants did end up including nurses, physicians, midwives, advanced practice nurse practitioners, as well as physician assistants. To sort of summarize the high points, and I understand that the organizers of this session will also share a brief of our survey, so many of you will have the opportunity to look at it in a little more detail. But our findings were that most clinicians recognize the importance of environmental health and know that environmental contaminants contribute to the burden of disease. Despite this, we found that most clinicians did not discuss environmental exposure concerns with their patients over the past year, and a good third of them have never had that conversation with the patient. We also found that less than 10% of clinicians have ever had any training in environmental health. In terms of barriers, we found that limited time and limited knowledge were perhaps the top 2 barriers to having those conversations. We also found that contaminants in food and drinking water were the least discussed contaminants for these providers. Most of the clinicians, about 76%, never provide any patient education material on environmental exposures, and we're not aware of readily accessible information to share with their patients. So knowing that clinicians lack the tools, time, and training, we're using these results to develop some training as well as patient and provider-focused outreach material.
As you reflect on this work, how do you think clinicians can move forward and become more educated about this topic? Do you have plans in that regard?
We certainly do. And I think we found that most clinicians do have a desire for additional information. They just didn't know necessarily how to access it So we're doing a number of things, including developing pre-licensure and post-licensure training specifically for nurses. We're developing pre-licensure and post-licensure training for physicians specifically, and we're developing outreach material for patients specifically in underserved communities in Massachusetts and for individuals that potentially live near contaminated sites.
Thanks. You mentioned that the results of your survey will be posted with the other materials from this session. I think, and Kathy Barry wisely suggested that the Wilmington study itself be posted. I think that will happen. It's a large PDF file that can be posted. A couple of other resources that come to mind for the clinicians on this webinar, or however we characterize this. The CDC has a online continuing medical education module or series of modules called The Story of Health. And one of those is about environmental exposures to children and the subsequent risk of leukemia. It happens that it's pesticides in that example, but that might be of interest as part of a longer series of chapters that the Story of Health goes through. And I'm sure there will be other links and references that people can add in as this session is put online. I'm going to switch now to some questions in the Q&A. I'm going to take the first one because it says, has Massachusetts banned or regulated TCE and PCE? And I think Massachusetts itself is governed by EPA's regulations with regard to TCE and PCE. Perchloroethylene is the dry cleaning solvent used for years as the main chemical in dry cleaning. And TCE is now, thanks to Kate Guyton, actually, is when she was at EPA, she led the effort for EPA to declare that TCE, trichloroethylene, was carcinogenic to humans. So the federal agencies really set the tone on that. Massachusetts doesn't ban or usually doesn't regulate chemical contaminants in opposition to what the federal agencies say. And I think in these two cases, TCE and PCE, they follow. These are very, in drinking water, both of these chemicals are regulated at 5 parts per billion in drinking water. One could argue maybe it should even be lower, but that's the way it stands now. NDMA in drinking water, I'm not aware of state or federal drinking water regulations. There may well be some. And in fact, Kate may weigh in on NDMA. Let's see if there's some other questions that I could field here. Oh, here's an interesting one. Do you find that social media has aided in community awareness and advocacy? I'm thinking about parent community chats on Facebook and other social media sites. Cindy McEwen has just said she's going to answer this question live. So take it away, Cindy. And I think
actually, no, I wasn't going to answer it. Sorry if I clicked the wrong button. I would love, though, to hear from Brian and Kathy on their perspective on that,
Great.
please.
Okay.
Thank you.
Yeah, that was a magic click somebody did. But anyway, do Brian or Kathy have a comment on social media and how it has aided or perhaps not aided in community awareness and advocacy in Wilmington?
It wasn't a huge player in our situation, from what I recall, Kathy, right? I mean, we were pretty established before social media was a thing. I do suspect that there's some challenges there around HIPAA, and it gets a little tricky, right? And I think that's why Kathy and folks that kind of started driving this thing is why they went to the newspapers to just put out a call out, right, to meet about it. The trick with social media is that, you know, it's property of whoever that platform is. So there's a bit of a privacy question there. I don't know how much I'd want to be divulging medical information on one of those media platforms, other than maybe just a way to coordinate and then you meet, you know, in person or over Zoom or something. That's what I'm thinking.
I feel as it would be, it would have an integral play in all of our, you know, the numbers of meetings that we had once those committees were established, and the transportation of the many from the Department of Public Health two, actually in the Kelly Hill Committee, they came out every couple months. And, so these remote types of gatherings, this Zoom would have been wonderful. We actually learned about our study over Zoom. And so I think it has a role, and I am kind of I think about all the numbers of hours spent in these last 20 years, how it probably would have been able to be cut back a little bit, but I think it would be a great way. And I think we wouldn't have been in the dark for so long had we had social media during the years that we were looking for everybody that was affected.
Yeah, we're in the Zoom era now. So actually it's a different century, so to speak, from where things started. In Woburn, of course, it was all community meetings. There was a citizens group called For a Cleaner Environment, which met at the Trinity Episcopal Church. And I attended some of those meetings actually. And that's where people really talked. And parents of children did come to those meetings. So they were willing to some extent, share their child's diagnosis, but it was really about organizing and the strategies about approaching the state and federal government. Actually, we wound up, getting a lot of help from Senator Ted Kennedy's staff, and then Centers for Disease Control assigned somebody to come and work with the state health department to conduct the first of a series of studies. So let's see. I have a question here for, are there resources we can direct patients to that feel they have a local cluster? One example is the fireworks site in Hanover, Massachusetts, and the large number of patients with brain tumors that grew up in that area. There is a resource for at Boston University School of Public Health for people who are considering trying to get a community health study done. It's on the Department of Environmental Health website. The person who manages it is Dr. Madeline Scammell, Scammell. So that is a place to go in Massachusetts, at least, for insight about cluster studies, the pros and cons. As I think those of you who will eventually take a look at the Wilmington study will realize that even though it was done at the state of the art and the water modeling was state of the art, it was still based on a fairly small, thank goodness, a fairly small number of cases. So sometimes small numbers result in what are called wide confidence bounds around whatever the risk estimates are, and a sort of inconclusiveness in the results. That's not the case, I think, in the Woburn investigation, but it took heroic efforts to get to the point where even with a small number of cases, they could say some useful things. And the cleanup, of course, is going to continue in any case, whether the findings were statistically significant, as the phrase goes, or not. So that's one of the warnings about getting a study of cancer clusters. They're often inconclusive and frustrating. I would say that it took a long time for the citizens of Woburn to feel like they were getting their questions answered. Some may still not feel like all of their questions were answered. And in Toms River, New Jersey, which was a larger study of childhood cancer, 42 cases, that was, again, with state-of-the-art techniques, and it was published about 20 years ago now. It wasn't published, it was released as a state report with massive input from the Centers for Disease Control. There are some parents who think, well, why wasn't there a stronger result from that? And I think the answer is typically these studies are hard to do. And when there are small numbers of patients, a little difference here and there as to who was exposed or how much they were exposed to can change the risk estimate at the end of it. Some people say epidemiologic study is like a lab experiment where you let the animals out of their cages and they all run around and you don't really have very good control over what they're exposed to. I have another colleague, former chair of my department at Boston University, who used to say that the definition of a public health disaster is something that's so bad that even an epidemiologic study can pick it up. So that's just kind of the reality of cluster studies, is they're not easily done, and the key is always, can you identify who's exposed and at what level they're exposed to come up with some kind of a meaningful risk estimate from that exposure at the end of it all. Let's see, I'm scanning through. Oh, here's a story, a question from a pediatrician from the Pittsburgh area, Dr. Ned Katayar. How do you plan to get educational information about environmental exposures to providers' inboxes when they may work for different competing health systems? Mark, you have an answer that might apply. Your answer might not apply in Pittsburgh, but tell us how you would approach that in Massachusetts.
In my experience through this work, we've found that if you've talked to one clinician, you've sort of talked to one clinician, and you need sort of a multi-pronged approach to accomplish it. I think some strategies that we think are bearing fruit is specifically with practicing nurses, which a lot of times have a lot of patient contact, sometimes maybe more than some of the physicians, is to work directly with labor unions. And we've done that with, say, the Massachusetts Nurses Association, and provided training on specifically environmental health awareness, have ensured that training, like this one, I believe, provides continuing nursing education and some post-licensure credit. Other things we're doing are also working with fellowship programs. For example, the University of Massachusetts Medical School has a preventive medicine residency program, and through those fellowship trainings, it's an opportunity outside of medical school training, where there is some formalized instructions that you can build in to the postgraduate training of physicians to incorporate it. There are some institutions, even within Massachusetts, some healthcare provider networks that have agreed to distribute our material. It's one of the ways we were very successful in getting a lot of pediatricians specifically to complete the survey by distributing it through the PPO network, a distributed network of pediatricians in Massachusetts. So I think our success is not to just focus on any one thing, but look for multiple channels and opportunities to get the information out. One of the best opportunities we have is when there is a community impacted by a contaminant in drinking water. Many times it's the school nurse or local pediatrician or a local provider that's reaching out. We always seize on that opportunity to offer them some training, to offer them information about resources for clinician training from ATSDR, from CDC, or others. So it's really that unilateral approach.
Great, thanks. And I know that Dr. Katayar is a member of Physicians for Social Responsibility, and I think Physicians for Social Responsibility, it's in some areas at least, has taken up this issue of environmental contamination and its human health consequences, and has training materials. The Greater Boston Physicians for Social Responsibility has those things, for example. Another question on the Q&A is from Suzanne Condon, who used to be a very high administrator in the state health department during the time of the Wilmington study. And she asks, is there a Q&A for the Wilmington study that could be posted along with the link to the full report? I will take that up with the organizers of this panel. I think it probably is something that the state health department either has done or could do. And if there's such a thing available, we will make that available to the audience here. I got a question or a comment, I guess, from Dr. Linda Birnbaum, another of our heroes in the field of environmental health. She used to be the head of the National Institute for Environmental Health Sciences, and she says, federal EPA is very slow to regulate. Often states move ahead of EPA, and I know that's true. The question that I was trying to respond to earlier was about TCE and PCE, and I think there are regulations, at least in drinking water, from the federal EPA on both of those. And the one about TCE is strengthened tremendously by the work that Kate Guyton did when she was there. But the point is still correct, that EPA is very slow to regulate. And even when it does, it takes, once the science is in, the time between the science being in and the regulations showing up in the Federal Register can be years. Let's see. Rachel Massey from the Toxic Use Reduction Institute in Massachusetts says TCE and PCE are both listed as higher hazard substances under the Toxic Use Reduction Act. Businesses that use large quantities are required to develop toxic use reduction plans for these chemicals. And that's a good point. The exposures that show up in drinking water typically come from some industry that's either improperly disposed of them or had leaks that they either were or were not aware of. So reducing the use of those chemicals like TCE and PCE is a way to prevent worse exposures than what otherwise have occurred. And the TURA Act in Massachusetts, we're blessed in Massachusetts that there is such an act. I think it was passed in 1989, and the use of toxic chemicals, including TCE, have come down dramatically as a result of the provisions of that act and the recommendations of the toxic use reduction industry. I mentioned dry cleaning earlier with respect to TCE. Actually, I think both were used. TCE and PCE were used in dry cleaning, but there are now regulations in place about where dry cleaning using those chemicals can be done, and whether it can be done where there are in a building where there are children living upstairs or near a daycare center, or in the same building as the daycare center. And I think California is sort of leading the way on this, but eventually those chemicals will be phased out as chemicals used in dry cleaning. There's an alternative called wet cleaning, which is available, which doesn't use those chemicals. And as that moves in and those chemicals are reduced, the problems of them showing up in drinking water will be less. There's a comment about a PEHSU, which stands for a Pediatric Environment Health Sciences Unit, I believe. And that is available to primary care physicians. Let's say, I'll read the comment. A PEHSU direct and PC, as a PEHSU direct and PCP, it's difficult to fit everything into a 20-minute appointment. The field of medicine continues to grow in terms of content, but training duration hasn't changed in many decades. Folks have been working on getting EH into medical ed for years. As I understand it, it sounds like a person who is a primary care physician, and have heard repeatedly, unless the subject matter is in the board exams, schools are not likely to add it to the curriculum. I'm sure that is true. And years ago, I used to give lectures to the medical students at Boston University Medical School about this stuff, about Woburn, and in fact, and it was like an hour and a half out of their 4-year medical education. So I don't know if they're even still doing it, but it was a tiny piece. Some students, of course, were very interested, but it doesn't necessarily rank high, especially if it's not going to be on the board exams.
Dick, if I may, the PESUs are an excellent resource that certainly this group would, I think, benefit from learning about. The Pediatric Environmental Health Specialty Units are a group of specially trained physicians, usually medical toxicologists, pediatricians, that are organized into regional centers and sponsored by the American Academy of Pediatrics and US EPA. We're fortunate to have one here in Boston at Boston Children's Hospital, and they are collaborators on our work, in fact. So this particular physician, who I suspect is a pediatrician, is raising an excellent point, and I just want to underscore the value of PESUs as experts in environmental medicine and reducing exposures amongst children.
Great, good point, thanks. Here's an interesting question for Kathy and Brian. How do you separate the emotional aspect of this situation from the search for evidence? Is there something that can be separated? Is that something that can be separated from your on-the-ground experience, either Brian or Kathy?
No, it can't be separated. People like somebody like myself who wasn't having to deal with the day-to-day issues of taking care of a sick child. It was fortunate that we had a number of residents that were in my category, that we could channel our efforts and get everything that we could get as far as data, as far as research, and yet we were still touched because we meet up with the families and tell them where we were and how, and I can't tell you how many times I went home crying that I wasn't able to move things forward faster. You can't separate it. No.
Brian, is that you?
Yeah, there's no separating them. I mean, to kind of build off what Kathy said, I think a big part of it too, that's the emotion is what drives you. The emotion is what keeps you in the fight. The emotion is what keeps you pushing to get the answers, to get the data. And then on the conversely, you know, when we got this study actually, you know, when it actually came out, the joy, joy seems like an odd word, but the joy of knowing that the efforts of this group made this data available to everybody that they could potentially use in other studies for other communities. People, that study is going to touch people in lives that we'll never know in Wilmington. And I think, you know, that human connection, even though it's an indirect connection and it'll have ripple effect over decades, I'm sure, beyond here, you can't separate the emotion. You just try to keep it in check when you need to and let it drive you and keep you running when you need it.
Well said. Well said. I'm seeing another couple of questions where it says that one of the other people on this background group will answer, so I'll skip over those, although one of them is from a friend of mine. Any cluster study in Westfield, Mass due to PFAS in drinking water? Mark, do you know about that?
Sure. There's not a cluster study per se that I'm aware of, but there is an ATSDR exposure assessment study. It's called the Hampden County study, and you can find information online about that study assessing exposure to PFAS in drinking water and some biomonitoring measurements. They're measuring PFAS in serum and in urine in that community.
Along with some other communities in Massachusetts, is that right?
That's correct, and around the country, but
right.
Westfield specifically is included in that ATSDR study. And Silent Spring Institute, incidentally, is also involved in an investigator-initiated study where they're looking at some health outcomes and PFAS exposure, and they'd be in a better position to talk about that work.
Right. It does raise another chemical of concern, which we haven't mentioned yet, perfluorinated alkyl substances, PFOA and PFOS are the most common ones. And that's, as Mark said, the topic of investigation around this country, around the world, actually. And there'll be more to come on that. As you probably know, it's the chemical that makes Gore-Tex repel water and used to make Teflon pans, not stick. Among other uses, and so it's so persistent that the half-life has been estimated, at least in one credible study, of 92 years. That means that it's really not possible yet. It's only been in existence less than 92 years, so it's not possible yet to say how long this is going to be with us. But it's in all of us. It's a chemical that's so persistent that it goes throughout the environment and therefore into all of us and other species. So that's for a topic for another day. It may eventually wind up being a topic for carcinogenicity. My Q&A for some reason seems to have frozen, so I can't scroll down any longer. Let's see if I can. Yeah, no,
Dr. Clapp,
I've got a couple to suggest. Go ahead, Richard.
Oh, I was just going to say, if you close the Q&A window and try to open it back up, it's possible it could come back. But Ruth Ann, go ahead too.
Yeah, go ahead.
Sure. I was just going to say there's a question, any outreach training for those who work in oncology who can, to help them understand and get better educated about environmental exposures and how they can report and alert about case clusters.
Well, I will say, in terms of the health department, our training is geared of course, towards primary care providers, but certainly specialty care providers, those in oncology, could certainly participate. And I think there would be some useful information related to that specific question. To build off what Dick was saying as well about his former colleagues at BU, the Department of Public Health also has within the Bureau of Environmental Health a group that responds to specific concerns about cancer within the community. So if individuals do have questions, for example, the Hanover fireworks site was raised, questions about cancer outcomes around a site, they can certainly contact the Massachusetts Department of Public Health, the Bureau of Environmental Health within DPH, and ask to speak to somebody about a community cancer investigation.
So Ruth Ann, I don't know if you have any other ones, but
Sure.
I'm still frozen with the Q&A, but I did
Okay.
get a message from Tim that the 2-minute, this is about a minute ago, the 2-minute warning is that we're about to wrap up. So I think with that, I will pass it back to the two of you and thank you all thank you to the panel. Thanks for those great questions. And thank you to the speakers for a fascinating session here.
Yeah, thank you. I'd like to echo that. The speakers and the panelists were just amazing, and I learned a lot. I know that the audience also learned a lot. And I think that it would be great if we can continue to have the conversation with the colleagues on the topics covered today and continue this dialogue through until the next session. So that next session will be on February 16th. The topic is air pollution and cancer, and the science linking air pollution to cancers is really strong, given that reducing exposure is often beyond the control of the individual. The session will focus on opportunities to address air pollution in public health, research, community settings, and roles that clinicians may play. So finally, if you wish to claim your continuing medical education and nursing professional development credits for this session, you must complete an evaluation. The evaluation will be available within 3 business days, and everyone will get a reminder email to complete that for those credits. And even if you're not claiming credits, please complete the evaluation and give us your feedback. You'll receive a certificate of participation. And I apologize, we couldn't get to all the great questions that were out today. There were really some very good ones. And so next time we'll make this session 8 hours long, not 2 hours long. So we really can go through the whole discussion that would be of interest to see. So one final thank you to all the participants and to the organizers for this. And I look forward to seeing everybody else on February 16th. Thank you.
Thank you.